Mosengo.
Your screen is not shared. Good morning.
Go.
Hello, everyone. Good morning. It's a great pleasure for us to have Professor Kambez Benam with us today and our e-seminar series on Translational Biomedical Engineering. And we're excited to hear about bio-inspired engineering systems for preclinical respiratory research that is going to talk about today. But before we start the presentation, as usual, I'd like to go over some housekeeping notes. First of all, if you have any questions, please make sure that you use the ask a question box, which is at the bottom of the screen, to type in your questions. Also, use the chat box on the right-hand side to communicate with the speaker, with us, and also with other participants in the event. Our next speaker will be Professor Kipret Mekwanent, who is a professor and a chemical and biochemical engineering at Western University in Canada. Make sure that you follow us on Twitter. Our handle is TransLBME. Also, you can follow me and Human on Twitter and LinkedIn to get the most up-to-date information about these talks. Last but not least, I'd like to thank Montreal TransMedTech Institute, who has been with us since almost the beginning of this e-seminar series. They've been supporting us all the way through this, like the 2 years that we've been doing this e-seminar series. So thank you, Montreal TransMedTech. So with that, I'd like to introduce our speaker today, Professor Benam, who is the founder of Lung Microengineering Lab, which brings together researchers from the engineering, biology, biopharmaceutical industry, clinical, and business communities with the aim of developing new technologies that recreate complex human organ pathophysiology in vitro. And then his lab applies these technologies to discover novel therapeutics and personalized biomarkers. His research specifically focuses on applying disruptive technologies that enable his team to elucidate cellular and molecular mechanisms that govern tissue pathology and offer protection during lung injury and host environmental interactions. So Dr. Benam received a bachelor's degree in Pharmacology from Newcastle University in the UK, and then a Doctor of Philosophy in Immunology from the University of Oxford in the UK. He was then trained as a Technology Development Fellow at the Wyss Institute for Biologically Inspired Engineering at Harvard University, where I met him for the first time, and then I was basically enjoyed a few conversations that we had about organs on a chip back then. Dr. Benam has been the recipient of multiple awards, including the Society of Toxicology IRSS, Baxter, and Lush Young Investigator Awards, as well as Science and Innovation Center Rising Star Award. And the list goes on and on. He has many publications and in high impact journals, and his work has received extensive press coverage by BBC News, Stat News, Harvard Gazette, Washington Times, and IEEE Spectrum, and so on and so forth. And then publication list is in leading journals, including Nature Methods, Cell Systems, and so on and so forth. He is also a co-founder on 12 pending patent applications and multiple reports of inventions. Dr. Benam currently manages about 10 million funding in his lab, and then his also company. He is also the founder of Pneumax, a biotech startup focused on development and commercialization of next generation living lung organomimetic systems, which is I think he's going to partly cover his work in the startup as well. So thank you very much, Professor Benam, for accepting our invitation, and we're very excited to hear your talk. So the virtual floor is now yours.
Do you see my slides?
Not yet. Yes.
Yes, great. Well, thank you so much. I want to appreciate the opportunity. I'd like to thank Professor Sawaji and Akbari for this opportunity and for everyone for their time. So thank you so much. What I would like to discuss here is kind of development of engineered systems, living and non-living, that they can fit in the preclinical space for the lungs and lung diseases, pathophysiology. And for full kind of disclosure, I'm a founder of Pneumax, as well as my academic appointment as at the University of Pittsburgh. But you might ask yourself, why lung diseases? I want to show you a graph from WHO, World Health Organization. They list the top 10 leading causes of death worldwide. And this data gets reviewed every 2 to 5 years. And this is how the plot looked like until the COVID actually hit us. And this hasn't really changed apart from the fact that respiratory infections has moved up. But what you can appreciate is that 3 of the top 5 causes of death in humans globally are lung related. Lung infections, chronic obstructive pulmonary disease, or COPD. You might know this mostly as kind of a disease associated with the human smokers, and lung cancers. They're actually a major burden on human health. One thing you might also notice is that the 3 of these kind of conditions which are debilitating are a result of 2 things. Either interaction of host, which is us humans, with a virus, with a pathogen, like influenza viruses, coronaviruses, pneumonial bacteria, or interaction because of us interacting as a host, interacting with what's in the environment. Mostly notably, I can say the case of cigarette smoke that leads is a prime principal risk factor for development of COPD, and is actually a major contributor to development of many types of the lung cancers. In terms of respiratory viral infections, I want to kind of emphasize beyond coronaviruses how actually a pressing need it is to have better model system to study those. This is a plot that actually, a table that actually illustrates the number of new respiratory viruses that emerged over a span of 2 decades up to 2014. So since 1995 up to 2014, we had about 20 different respiratory viruses that have emerged. Half of these viruses are influenza. You might remember kind of swine influenza back in 2009, which kind of spread globally. We were fortunate that it did not have as much of pathology as one would have expected. But on the other hand, we also faces other pressing respiratory viral infections such as SARS-CoV-2. So in other words, we are living in a system that we're constantly challenged by the pathogens that's in the environment. And among those viruses, respiratory viruses, still pose a huge burden on human health, as well as on our society and economies. The other aspect is something that does not necessarily kill similar to kind of many respiratory viruses or smoking, but is actually something that initially came into the market as a kind of a safer alternative to cigarette smoking, but has quickly picked up its appeal among the youth. And that's electronic cigarettes, or which formerly known as electronic nicotine delivery systems. In the United States alone, almost 1 in 4 or 1 in 5 high school students has tried in 2020 a tobacco-related product, and of this, 1 in 5 has used an electronic cigarette. So it just tells you the scale of the fact that these are people, often many of those are first-time vapors, so they will never have a history of smoking that they try to replace it. They try to come in, because a lot of these products emerge very quickly, and they need to be better kind of understood in terms of pathology, toxicity they have, so they can be better regulated. But they have been emerging at such a astronomical pace that it's actually very hard to keep up. So society has actually owes to citizens to be able to better understand what sort of pathology it causes, what is the safety profile, and how we can actually better regulate it ultimately from a public health standpoint. So we have kind of these pressing problems, i.e. leading cause of death in humans, lung-related or societal challenges such as emergence of electronic nicotine delivery systems. But what are the model systems that outside the human, in a preclinical setting, we can actually use and study those? I want to start by going to in vivo models, or animal models that are commonly used for a lot of respiratory viral infections: rodents, mice, and rats. One thing very interesting about them, many of them are not laboratory mice and laboratory rodents. Rats are not the natural host for many of the human influenza viruses. Even similar to coronaviruses, they're not naturally susceptible. So you need to either make it transgenic mice, or you need to evolve a virus to infect the mice. Which in this case means that you actually might have a different virus than what you originally came into and cause an outbreak, whether it's an epidemic, it's an endemic virus, or it's a pandemic. Now, what are the next model systems actually that people actually use? It's not as common, but ferret model is used. But the sort of challenges with them is that the drug pharmacokinetics and safety is different than humans. And plus, we don't have as many reagents to characterize the disease biology in ferrets compared how to people can do it in mice. And the next kind of elaborate animal model is the guinea pig, and they have attenuated outcomes. So in other words, if you want to mimic, for example, in the context of influenza, severe outcome, a severe pathology, same as, for example, the avian influenza, H5N1, with a guinea pig, you might not be able to do that because they naturally cannot develop that outcome as we would expect it. So that means that these model systems are not scientifically suitable. And plus, obviously, there's a huge ethics about use of animals in the clinical setting. Now let's look at the in vitro models. In vitro models, when you want to study a human lung, and often I'm talking about the conducting airways, you either use cell lines. They may not be anything related to lungs, like 293 T cells or MDCK, or they might be actually from the lungs, and they might be cell lines, or they might be primary cells that were just isolated and cultured in vitro. What is the challenge here? Because they don't have the differentiated phenotype, the microarchitecture that you have in a conducting airways. What you would see inside a conducting airway is a kind of mucociliated epithelia that line the conducting airways, and that's what it gets exposed on the rhythmic breathing to what you inhale, whether it has the pathogen, whether it has the allergen, whether it has the particulates from the vaping or from smoking or whatever that it comes in that's exposed. So this model system, they don't have those cells similar to microarchitecture what you have because they are in a de-differentiated format. The next advanced model to that is actually people use a system that's called transval insert systems. So this is a microwell plate or milliwell plate that inside there are inserts, and the bottom of the insert has a porous membrane. What are the kind of the challenges that associated with this, whether you want to study influenza infections as a respiratory viral pathogen or even cigarette smoke? They don't often mimic the organ level or organ-like pathophysiology. This is a more complex model because you can create some aspects of epithelial cell microarchitecture, but still you cannot create a multi-tissue kind of system that you can study the crosstalk between the tissues and cells in that system. None of them can enable you to study dynamic interaction between lung cells and circulating immune cells. What happens during an infection, when you and I get infected with a virus or a bacteria, when we have a sore throat, when we have when we get a viral infection, the circulating immune cells sense something is kind of causing inflammation and causing injury in your lungs, and they home there. It's never like that actually someone comes in and dump a bunch of immune cells onto the lungs and say, hey, I want you to do what you need to do. No, it's not like that. So actually we have a dynamic processes that cells through circulation sense it, what's happening in a peripheral organ, and they want to home to that. Now, neither of these platforms can mimic aerosolized delivery to a pathogen, to smoke, to vapors, electronic cigarette vapors on their rhythmic breathing, and they're not actually well suited for studying real-time analysis what happens in a crosstalk between cells and organs, as I mentioned. Now, what about inhalation toxicology? I mentioned about e-cigarette vaping. So what are the model systems out there? Again, rodents, particularly mice, are very have been traditionally kind of a well attractive model for many investigators purely due to ease of access and the fact that they can study the whole organism. In vitro models, as well as a very basic kind of chemical physical analysis, and more recent in silico approaches such as computational toxicology. So from a scientific perspective, every of these platforms have an advantage, but we need to know the limitations and we want to do them. And of course, we need to take into account the ethics about this, particularly about using animals. So when you use an animal, it's like same as using a whole organism. That's the benefit, that you have all the organs there. Okay, so that's an advantage. When you use it in transval insect culture, you have a mucociliated epithelial. When you use endpoint assays, you have they are very easy to perform because you can measure concentration of something in the vapors or in the e-liquid. And computational methods, they actually have emerged as a very lucrative path to kind of replace use of animals. Now, the sort of challenges is that mice, rodents, they're obligate nose breathers. So when you want to inhale cigarette smoke, electronic cigarette vapors, they actually passively intake it. Unlike humans that need to actively take it, they actually take passive. So they take secondhand. Secondly, they have such a complex nasal turbinate, which means that a lot of particles they inhale is actually captured in the nose before it reaches the lung. So you often need to do long exposures or extensive exposure to a lot of particulates to see any biology effect. And plus, the histology in the lungs, the anatomy, immune system is quite distinct and different from humans. Translational simple models, I just mentioned some of the kind of challenges with them. And then when you do endpoint analysis, these are endpoints, so you cannot do in a real time, and they just done in silo. We want to see, for example, a new e-liquid came out for an electronic cigarette. What does this flavoring does? We just do chemical composition of that. It doesn't tell you much when it's done in silo, when you don't couple it with physiology or what actually the disease that actually can cause in the bodies. And with the computation aspect, the challenge is extrapolation of data. So the way we approach this, given this kind of pressing needs and challenging preclinical kind of platforms, we say we do two ways. We do a living system approach, which is we develop organ-on-chip technologies by bringing respiratory biology, tissue microengineering, and systems biology, and the non-living system, which is our kind of robotic aspect, to kind of emulate what humans vape or inhale as part of exposure to the environment. And we mimic something that's called vaping topography. That is, the parameters are associated with the vaping. So basically, when a person vapes, this is a science on its own. They take a puff. It's called puff time. The volume it takes in is called puff volume. Then they mix it with the tidal volume, or more or less. Then they inhale it. Then they exhale it. Now, until they take the next puff, there's a period that the subject, inhales and exhales normal air. And that's called interpuff interval. So all those parameters, they call vaping topography, similar to the smoking topography, which how people do it. And clinically, these are actually parameters that can be measured by using a transducer at the beginning of a vaping device or cigarette smoke. There's breathing mechanics. So basically, there's a pattern of breathing. So basically, this much of volume of air comes in, this much of volume of air goes out. But there's also a rhythm with that. Why does it matter? Because you can mimic the breathing with a healthy status, which is, say, an adult healthy subject inhaling, exhaling at the resting state about half a liter air in and out. You can mimic someone who has obstructive lung disease, such as asthma, COPD. They actually have problem exhaling. It's more difficult. Or someone who has restrictive, which is difficult to inhale, like someone who has more difficulty inhaling it. So when we create the mechanics of that, it allows us to kind of bring it into play when we want to emulate kind of those exposures. And then obviously engineering, which in a sense we do all aspects of it, from electrical, mechanical, and software, bio and tissue engineering. So organs on chip, microphysiological human small area on a chip. A lot of you have seen example of organ on chip. This is just a representative image. This is a PDMS device, has 2 micro channels. You can see inside it, they're all filled with the dyes, and the center area, they actually overlap. And the material here is PDMS, or silicon rubber, which is transparent, gas permeable, has its own advantages, which allows rapid prototyping, and disadvantages, which kind of can have adsorptive properties. But we started this kind of process of making a human lung small area on a chip. This is how the platform looks like. So this is a cross-section of the small area on a chip. And the reason we picked up small airway, which is defined by airways, conducting airways at generation roughly about 8 to 16. So from trachea, consider this generation 0. You bifurcate, you keep branching. You go to the right lung, left lung, primary bronchi, then it goes to different lobes, and it keeps branching. You roughly have up to 20, 25 generations of bifurcation that happens. Then it becomes alveoli, which is kind of your air sac. That's where kind of there's expansion and retraction. You have the air, kind of the oxygen gets into the bloodstream, and the CO2 kind of came back, and we push everything back in here, okay? So small airway generation roughly from 8 to 16. They're defined by diameter of 2 millimeter or less. Why they're important? For long, they actually were underappreciated for their role in disease development and pathologies. But later, people realized actually a lot of pathology development happens there. And because the collective surface area is so much, if they get engaged and they get involved at whatever capacity, which basically whatever proportion of them get involved, it can impact the airflow, how much air you can actually bring it down to the alveolar or take it out. So the remodeling, the inflammation, infection on the inner surfaces and the walls of the small area is particularly important. So to mimic that, we built a small Airway-on-a-Chip. This has two compartments, two microchannels. The top has a height of 1,000 micron or 1 millimeter, and the bottom has a blood vascular channel which has a depth about 100 to 220 or 20 or 50 micron depth. We wanted to culture primary human-derived small airway epithelial cells, these bronchial cells, and guide them on the air-liquid interface, which is air-blood channel, to guide them to become mucociliated, which means they have this hairy structure, the cilia, and they have mucin-producing capability. They also have other epithelial subtypes, which they are differentiated, but they are like air, exactly what you have in the body. And they're separated by a porous membrane from endothelial cells that are lining underneath them. And endothelial cells, we use lung microvascular endothelial cells, primary human lung drive. And then we had a vascular shear stress of about 1 dyne per centimeter square to roughly mimic what happens at the post-capillary venule. Why? Because that's a major site of, that's one of the primary kind of sites of recruitment, adhesion of immune cells to the periphery for during infection and inflammation. So we developed the model. We had to characterize it. We have done this with so many donors, and I can tell you this is quite a low throughput platform. So it actually takes so much, but it's high content and high value because you can capture a lot of data when it's built and when it's successful. So you can see the ciliary active ciliary beating on a profile image, and you can see the cilia. And what you see here is more or less the microvilli on top of the other non-ciliary cells that you have. They have a pseudo-stratified structure, so they're not exactly one layer. They're not exactly multi-layer, multi-stratified. They are actually pseudo-stratified. That's what happens in the actual our conducting airways. And we stain them for other epithelial subtypes, such as mucin, which is mucin 5AC. And they form the passive barrier, which is the tight junction to kind of protect us from outside. That's passively protecting us from outside. And then the endothelial cells on the bottom, you can see the nuclei here and CD31, the pan-endothelial cell marker, as well as selectins like E-selectin, which is important for interaction with the circulating immune cells. So we developed this platform. We validated with 10 to 15 donors, 2 to 5 biological replicas at any given time, with so much of kind of actual resources and time that takes to take this. Now, who cares? We need to create value to that. The reason we did it, we went back one step forward. We said, okay, the reason is that we have all these clinical challenges because of respiratory diseases. We want to have better models so we can apply them ultimately. So the translation aspect of it. And in this case, the way we did the translation is that we use COPD as an example. As I mentioned, COPD was the fourth leading cause of death globally, and actually in the United States is the third leading cause of death. So that's actually a major burden on public and a major burden actually on the healthcare systems. So what causes a lot of problem for the people who have COPD is actually acute exacerbation. So generally speaking, it's a progressive disease that progresses and gets worse. It does not stop. We don't have anything to cure or stop the disease progression. But what causes a lot of burden for this patient is actually when they get exacerbation. So for example, during flu season, or even sometimes when there's other kind of infections around, when they get infected, they often, not all the time, they often do much worse than actually a normal subject. And ordinarily, someone that is, you can think about sex-age match that it doesn't have COPD. Why? Because they have dysregulated and exaggerated inflammatory response to their exposure to that pathogen. So we said, what if we try to make a COPD lung Airway-on-a-Chip to mimic that and apply this platform for therapeutic testing? Since there's no treatment for exacerbation, really, it's more palliative, and common treatment is actually people use steroids, and we use budesonide as a kind of a standard of care steroid that's given in clinic. And we wanted to compare it with an experimental lead drug from our pharmaceutical collaborator to see how they compare in preventing recruitment of immune cells to the lungs. And to look at that, we said, okay, the two major innate immune cells, which means the very first immune cells that they come into the peripheral organ and tissue during infection inflammation, in the context of COPD exacerbation, are macrophages and neutrophils. Neutrophils are the most abundant immune cells in our blood, and they can quickly mobilize, and that massively produced in high quantities in the bone marrow and can quickly mobilize in response to the cues they receive from circulation. Whereas macrophages, they can be fetal macrophages that they actually stay with us or in the patient throughout their lives, or they come derived from monocytes that were recruited to the lungs. But that process can take several days. Neutrophil recruitment, and actually they they capture to the kind of peripheral tissue, is actually a very quick process. So we said that we know that these two correlate with the pathology, so why not studying with the neutrophilic inflammation? So this is how we studied this. So we made the COPD lung airway-on-a-chip. This is the airway channel, this is the blood vessel channel. We populated the airway side with the COPD-derived airway epithelial cells. We differentiate them to become mucociliated, like how they line in the body. We co-culture with them with the microvascular endothelial cells on the surface, and then we challenge them on the luminal surface of the airway epithelium with toll-like receptor 3 ligand, polyiacid. This is to mimic viral infections. We did not want to choose, for example, rhinovirus over influenza virus or over a coronavirus, not this coronavirus SARS-CoV-2, but seasonal coronavirus. So we did not want to choose one because it's very hard to stratify the patient based on which country you look at. But if we say we broadly mimic viral infection, we might have a better insight to that. So we mimic that, and we circulated neutrophils that are isolated from human blood and resuspended at the densities that you have in the human blood, basically in the same number of millions of cells per mil that you have in the blood, and then circulate them at a vascular shear stress of 1 dyne per centimeter square. This is how the dynamic interaction looks like. So these are neutrophils. There's a lot of them passing by, and you can see they scroll and tightly adhere to the endothelial cell. You can see autofluorescence of underlying endothelial cells here. So we made this a dynamic interaction to see how we can use this in the context of drug treatment. So we had an exacerbated COPD chip that received no drug, or received, in other words, received the control, which is placebo in this case, and was DMSO that contained the drugs. We can see a lot of neutrophils adhere, so every grain is a neutrophil. Now, when we give budesonide, a steroid, which is used in clinic, we can see that there's still quite a lot of neutrophils adhere. And that's both interesting, but also disappointing, because you would assume they can actually help with a lot of inflammatory processes in the patients. But when you look at the BRD4 inhibitor, our experimental drug, we found actually it actually reduced the inhibition, it reduced the binding of capture of circulating neutrophils to inflame endothelium. So that made us to repeat the study with so many donors, and we found actually that it's actually genuinely true, because when we repeated with as many donors as we could, which is in the order of 5 to 7 with 2 to 5 biological replicates. And remember, every experiment takes about 6 to 8 weeks just to do every single experiment. We found this is consistent, that neutrophilic recruitment was only inhibited with the BRD4 inhibitor, whereas budesonide did not reduce that. In fact, it actually slightly increased. It was not significant, but slightly increased. We even studied increase it. So my point out of this here is that we can use this platform and study that how translate this technology to see how we can test lead compounds for the efficacy in a model system using patient-derived cells and more accurate kind of organotypic structures, and using mechanical cues such as the vascular flow to study which drug could be a lead, or more important, to kind of take it forward in the preclinical setting. Now let's actually switch gears. I focus on the living kind of engineered systems, which was an organ on chip. Now I want to talk to you about the bioinspired robotic systems, and this is the platform that we recently kind of published last year, and that's about making a robotic system to mimic inhalation exposure to the vaping products. And as I said, the need for that was because we want to see when someone changes, a company changes, and they add all these different variety of different classes of generations of the vaping devices. They produce a lot of features. They create all these features. You can change the kind of, for example, the voltage, the wattage, you can change the resistance, and obviously the user, one of the reasons they like it is actually the appeal it generates, because okay, you have more denser vapes that are coming out, you have more puffs, you can take it out of it, or like it can last longer, plus thousands and thousands of flavorings that's out there. So we said, okay, what if we develop a kind of a bioinspired robotic system that mimics inhalation exposure, mimics the mechanics of the breathing as well as the vaping topography, to see when the change is made, how it impacts the number and profile of aerosols that someone inhales into the lungs. Knowing that, we're making this assumption that the more of a particle that someone inhales the lungs, more likelihood that they may develop kind of a lung pathology, and i.e. more toxicological implications. This is the rationale, and this is how the robotic system looks like. It has multiple components. The very first component is the whole casing, which collects and puts everything together. What it does, it's similar to an incubator, cell culture incubator that you use in a BSL-2 or BSL-3 laboratory. The difference is that we had to make a system that has all the pneumatics and control and pressure-driven system to, one, take out any unused or any exhausted smoke or vapor from the system, so we can connect it to the exhaust of the building through a fume hose so we can take it out. Two, it can control the pressure within the system, because the system needs to also inject it with the carbon dioxide, same as a CO2 incubator that we have in the laboratory. It also needs to control for the humidity to make it as similar to what you have in, more or less in the mouth, just before you get full saturation, that kind of the particles before enter the lungs. You also want to kind of control it, because as soon as you start creating negative pressure to take out the vapor, unused vapors from the system, you also create negative pressure. You don't want the system to start become like in a vacuum to take pressure from all angles. So we had to create that, which is all the parts which is behind it. Then inside it, we have a breathing emulator, which mimics the inhalation and exhalation using the gas-like cylinders go back and forth. That's the rib cage, basically the mechanics of your rib cage. It has the vaping robot itself that creates the fresh vapor from electronic cigarette, and by changing the mouthpiece and using adapters, we can try literally any electronic cigarettes and any generation of ENDS device. On top of it, we have a dilution robot. What the dilution robot does is actually introduce the tidal volume you need to mix with the puff volume before you take it into the lungs. As I said, no one takes just a puff and that's it. They take a puff, there's a very small pause, they mix with the air, then that's how they take it down into their lungs. So that's what the dilution robot does. And ultimately, between the breathing emulator and the dilution robot, after the puff is generated, there's something we call it a sensor. So we use a laser-based sensor to kind of use the light to characterize the number of particles and the size distribution of particles as they go past through that. So where do we want to apply it? So we build this platform. Where do we want to apply it? So we did our homework, and we came to pick up this pathology, which is called EVALI. EVALI, or electronic cigarette or vaping product use associated lung injury, is a mystery lung disease that came out August 2019 in the US. It puzzled a lot of clinicians because they were coming and they were kind of ruled out, excluded from many other pathologies that were associated with them. But they had one thing in common. Those people, many of them had history of vaping electronic cigarettes that they contained vitamin E acetate and or cannabinoid products. So we picked up vitamin E acetate because that's very oily substance. In fact, we use vitamin E acetate as in ointments, in creams, facial creams, etc. We think it's a safe, which is actually it's essential it's an important vitamin. And it's actually important for what application we have, but maybe not good for inhaling it because you need to heat it up and it can impact what someone inhales. So we wanted to see if we can find a relation between vitamin E acetate and the number of particles and the profile of particles that one inhales. This is just showing you some statistics about this disease, EVALI, in the United States, that kind of based on the data that kind of how quickly we had a period of in 2019 from August when the cases went escalate up to December. Recently, we have a pick of these admissions because of this in the hospitals. So we started mimicking the kind of this exposure to a vitamin E acetate containing PGVG. PGVG is the base of e-liquid. So a lot of e-liquid that people use, or companies they sell, has a PGVG. Or propylene glycol vegetable glycerine, that's a mix together, and that's where they add all the other additives. Often they add things which are proprietary. Sometimes they don't add it. They actually claim what they have inside it. But one of the things we knew is that let's start with a simpler system. We have a very clear kind of base, which is PGVG, and we add to that vitamin E acetate at escalating doses to see how it impacts the particles that one would inhale. And we mimic the vaping topography and an average healthy adult breathing with a tidal volume about 500 and about 10 to 12 breaths per minute. And this is how the plot generally looks like on a simple PGVG. We quantify 4 fractions of sizes: 300 nanometer to 1 micron, 1 to 2.5, 2.5 to 4, and 4 to 10, all in real time. And you can see that the majority of particles actually are very small particles that exist from particles coming from a simple PGVG. So there's all a lot of particles that are coming out. Now, we added escalating doses of vitamin E acetate. You can see here from 1.25, 2.5, and 5. But you can appreciate the number of particles inhaled goes up. And even at the very small quantities, ratios of 1.25%, there was enough to see an increase in particles. The 4 graphs are basically the same. The only difference is that I'm showing you the plots for different particle sizes: 300 nanometer to 1 micron, 1 to 2.5, 2.5 to 4, and 4 to 10. So there's escalating doses. Now, if we put it together for a representative vaping session, so we say that the person did 7 times during the day, vaped, and every time took 9 puffs from electronic cigarettes that had either no vitamin E acetate or these doses of vitamin E acetate. What you can appreciate is that the number of particles goes up, and that's evident even as early as 1.25% addition to that. But what is interesting, the percentage of the particles is also changes as we add vitamin E acetate. So in other words, from 85.4% for the very small particles, we go down to 82.2, and the number of larger particles actually goes up. So we can use this platform to study not only that how addition of an additive such as vitamin acetate impacts the particles and the quantities that go into the lungs, but also distribution of particles. The distribution helps us to better predict where they could actually precipitate or what could be the biological consequence. We can kind of make assumptions based on that. We did repeat these studies with and without a nicotine. So you can see in the top left 4 here, you have no vitamin E acetate. Top right, you have vitamin acetate. And then we had increasing strength or doses of nicotine that people actually might use. In fact, these days, when we did these studies, we thought that 2.4% nicotine was the highest strength, but now we are surprised to see some companies sell actually at much higher doses. What is interesting is that nicotine itself had a minor impact that seemed to reduce it. It does not mean it's safe because we have not measured quantities of particles below 300 nanometer. But when we added the vitamin E acetate, it actually counteracted any effect of the nicotine. So in other words, vitamin E acetate was so strong that it still could increase the number of particles that one would inhale even when there's nicotine in their vaping device. And this is, again, a plot of the total particle sizes, as well as when you look at the percentages, you can see the distribution of particle sizes. Now, what is very cool about this platform is that we can mimic the breathing of someone who has a obstructive or kind of respiratory disease. So as I said, obstructive disease means, like, for example, asthma, COPD, and restrictive, for example, means IPF, idiopathic pulmonary fibrosis. So how this looks like in clinic. So someone in clinic, when they breathe normally, this is a representative image of flow volume look, which means that the spirometry, they do a test, they can tell you, okay, you don't have any, for example, you can rule out having obstructive respiratory disease. Now, someone who has, for example, asthma, obstructive, this is how the profile of the breathing looks like. So you can see the change in the flow rate and the volumes. And someone who has restrictive is actually, there's a shift here. So we said, okay, now to translate this for our robot, to mimic that mechanics of the breathing associated with lung diseases, we convert this to the breathing cycle. So we convert the flow volume in MATLAB to flow volume over time. That actually creates this plot. So we can actually mimic this breathing. And as you can even see, even with the obstructive restricted disease, the amount of air that they can take in is even less compared to someone who's otherwise healthy. So we repeated the studies without or with vitamin E acetate. These 2 on the left, they're normal breathing, normal adult breathing. The 2 in the middle, someone who has, for example, asthma or COPD, obstructive. And the 2 on the right, someone who has, for example, lung fibrosis, IPF, because they have a restrictive lung disease. What you can appreciate is that all of them have the same trend. So in all of them, vitamin E acetate increases the number of particles inhaled. That should not come surprising. But now, when we plotted the total particle count over the representative session, what we could find, the condition that had the highest magnitude of increase was the restricted. So in other words, someone who has restrictive lung disease shows a higher magnitude of inhaling more particles into their lungs after kind of inhaling a compound such as vitamin E acetate that causes number of particles goes up. So we could get insight through the physical profile of particles inhaled by someone because of the disease background, disease mechanics of the disease breathing. So just putting everything in perspective, I'm still within time. I want to kind of just recap this, that I hope I've been able to show an example of how we can use small Airway-on-a-Chip as a representative organ on chip to mimic some of the tissue aspects of the crosstalk and immune cell recruitment and application of that for a disease setting. I mentioned that the vitamin E acetate, VEA, has been strongly linked to the outbreak of E. valley, and we developed this kind of non-living robotic system to inhale and mimic that kind of exposure to vaping products. And we used this, and we found that vitamin E acetate, even a small dose, was sufficient to increase the number of inhaled particles into the lungs. And we hope that this platform can be applied to better understand that kind of as more vaping products comes out, how we can better understand the potential to cause the increase in number of particles that one would inhale, so we can better kind of protect the public by kind of understanding those potential in those new vaping products. With that, I'd like to thank you all for listening. This is my lab. Our funding agencies, US FDA, from our University of Pittsburgh, Cynic Institute, Department of Defense, Lush Prize, Moore Foundation, and NIH, NIBIB, and IEHS. This is my email, and for those of you who are interested in position in my lab, I'm hiring. Just let me know, and I'll be happy to take any questions.
Thank you so much, Professor Benam, for your inspiring talk. We really enjoyed it, and we learned a lot about the advances in this field. So I encourage our audience to put their questions in the question and answer box on the bottom of the screen. I think we lost Professor Benam.
I am here. I don't know why my video just disappeared. I apologize. I'm trying to figure out how I can bring it back.
No problem. So we got a question from Professor Blaszczuk, one of our previous speaker. Once we were talking about the Lung on a Chip platform, is wondering if you also include the fibroblasts inside your platform.
Yeah, so that's a good question. Basically, in other words, how much we care about including the stromal cells into what we model. And we are working on that. I think that's an area of interest to us. We have not done it yet so far, because I think we cannot just have an stromal cells without the stroma. We need to have the matrix there. And to have the matrix, you need to think about the structural integrity over time. You need to think about what aspect of physiology it's important to model and mimic. For example, a fibroblast from a disease setting like fibrosis, lung fibrosis, behaves very differently compared to healthy. Now, if you get those cells from a disease subject's lungs, you culture them, but you tell them not to chew the matrix, not to digest the matrix, because you want to keep a structural integrity. That is a problem. If you culture them in a 2D culture, just purely for example, on a membrane alone, that's a problem. So I think there's a lot of aspects of it we like to take into account, but it's essential that future we include those.
Perfect. Another question from Professor Marek. He's wondering, are you able to couple the Lung on a Chip platform with the robotic wrapper?
Yes, we are able to do that. That's actually one of the things on the future directions, because we want to show that an increased number of particles also translates directly to increased pathology at the site of exposure. So that's one of the things we will do in the future. Yes.
Okay, and the follow-up comment from Professor Blaszczuk is, they are the cells that cause fibrosis. Is that right? I think we lost Professor Benam.
I think it's back for some reason.
You're back. You're back.
Yes,
Did you
yeah.
hear? Did you hear the follow-up questions from
No,
Professor
no.
Plaschek?
Can you repeat the last question?
Yeah, he's wondering if the fibroblasts, the cells, are causing the fibrosis in the platform.
So in the platform that I showed mimicking the restrictive breathing, we don't have any living cells or tissue there. It's purely mechanical, kind of electromechanical system using an optical based sensor to mimic exposure to a vaping product and quantify particles that is inhaled. So think about it. We put a sensor as if in exactly at the trachea of someone who inhales a vaping device. We say we want to quantify number of particles they inhale, and we want to characterize distribution of those. That's exactly what we did. So we are here. We have not done any exposure on this with this platform. So Lung-on-a-Chip is not being connected to that platform yet. Now, why we say it's restrictive? Because restrictive is the mechanics of breathing. You change the breathing, you impact the airflow, the volume, all those sort of parameters that brings in and out of the lungs. That's what I meant by kind of emulating those parameters.
Perfect. I have a question also about the Lung-on-a-Chip platform. So you mentioned that the material is PDMS. So I'm just wondering, when you culture the cells, you have a monolayer of the cells on top of that, right? Epithelial cells. And so can we call them 3D still, or?
So when we culture the cells on the epithelial cells, I'll give you an example. On the kind of the porous membrane, whether it is a rigid membrane like polyester, polycarbonate, or PDMS membrane, they don't just stay flat. They start after air liquid interface. They form a pseudostratified structure. So from a flat structure, they kind of still tightly packed. They grow and they kind of grow on top of each other, pseudostratified. Some have cilia, some mucus producing. So that pseudostratified structure itself is in a sense a 3D model. Now, the 3D real comes into kind of bringing the other kind of cellular tissue components. So when we have the endothelial cells underneath it and we circulate immune cells, that is a 3D system. That's not anymore like a 2D or 3D. It's a dynamic 3D system.
Perfect. Thank you so much. And how about the mechanical property of the membrane? Because they're rigid, right? Or PDMS is kind of far from what we have in the lung.
Yeah, that's the holy grail. We want to kind of you want to kind of model something that happens in the body, but how much of what you model in vitro is actually mimics what's in vivo. So I completely agree. I don't think the material itself is close to what we have. Having said that, we still coat the material with a very thin kind of collagen matrix, just to make sure the cell, when they adhere, they're not seeing it just an external material. They're still comforted by having a kind of an ECM cushion to sit there. I don't think that lasts throughout the whole culture because they remodel it, they kind of change it, and they might even stick to that. But the stiffness can play a role. How important it is for epithelial cells, for example, I don't know. For endothelial cells, I think it's important because it still allows a lot of the adhesion that happens. If you look at even people generally talk about the fibroblasts, you put them in a flask is different than when you put it on a gel. That's very clear. But now if you do the same with endothelial cells, you can also sometimes see differences in how the cells adhere, etc. So back to your question, it's an important thing we need to tackle. I mean, it's not, I think as a community, we all need to tackle what material we can use that has closer kind of rheology, matrix rheology to what we have in the body, but it should be at least a doable platform. If we make it too complex, there's always this danger. We may not be able to make it, If we set the bar extremely high, we want everything to pass. It's sometimes very hard to do it. But as a next step of follow-up, I think it's important to be tackled.
Another question that we always get in the community when we submit applications is that, okay, we have this individual on a chip platform. How come, we can kind of see the systemic interaction between upstream, downstream? They are not complex enough to really mimic what we have in the body. I mean, immune system, circulation, and all these upstream, downstream tissues, they kind of criticize us, that we are really simplifying things.
So, okay, so I tell you my opinion about it. I think kind of the virtual human on a chip or virtual multi-organ on a chip system, that's the biggest step forward. But all comes down to the questions, all comes down knowing the limitation and the strength of the model we're using. So in other words, so let's say you want to connect two organs together. If you know the limitations are X, Y, and Z, but you don't address them, and you know that those limitations doesn't allow you to address your question of interest, that's a problem. But if I want to say, for example, connect an immune organ to the lung, just an example, and I want to see the crosstalk between those, and only I'm studying and know that given that it's far from complex of a human, what happens in a human, but all I'm interested in is that the mobilization of an immune cell out of, say, for example, bone marrow to the lungs or the other way around, to happen that's that platform, and I have the architecture in the both organs, and I know exactly what specific question I'm trying to do, that might be a step closer. But if I claim that I mimic the whole communication of human relevant communication between a lung and an immune system, that's a lie. I mean, you shouldn't even take me serious. So I can tell you that. So it all comes down to questions and knowing the limitations of every platform. At the end of the day, even organ and chip, I'm very critical of our own work. It's not a perfect model. It's a model. It's not a perfect. The perfect model is a collection of human subjects from desired population, not even one subject, that you want to test something, but it's never possible. You cannot say, I want to just give somebody this drug or test this on those people. You need to test them before you take it to human. So no model is perfect. Everything is relative. When the models complement, that's where you create value.
Exactly. Thank you so much. That's a great answer. So I pass to Mohsen. We have a couple of questions for Mohsen.
Well, thank you very much, Kambez. I don't know how much time you have, Kambez.
We've got like 7 minutes. Yeah, that's good. Yeah.
Okay, that's good. So my question is mostly about the application of these organs on a chip systems. Let's say the lung on a chip, and then how they can be integrated in, let's say, ISO standards that are acceptable by the FDA. So have you had any discussions with the FDA about, using these platforms for drug screening, for example, process or applications?
So I'm part of a tissue chip consortium, which is kind of run by NIH in the States. And there is basically a strong force, which is kind of, they try to bring all the kind of at least key players in this field together and say, hey, we have people doing different research. We want to kind of know each other. We also have an opportunity to engage with kind of different stakeholders, including FDA. How much FDA is taking this forward, etc. I think there's a lot of interest from community, from people, including kind of any engineers or any bioengineers, tissue engineers. They're interested in that. What the practical kind of output is going to look like, I don't think I know, or kind of I have any control over that, because that's a lot of decision making that happens that's kind of irrelevant to this. But to get there, I think we need to have trust and validation in these platforms. Without enough validation means that if I give you the same kind of airway chip, you should be able to repeat it. Validation means that many people can repeat the same data. The other thing is that knowing that what it can answer, what it cannot answer. So, for example, some of the pathologies relating to developmental diseases, it might be very challenging to recreate them with a organ on chip model, including lung on a chip. So we need to know, maybe there, organ on chip cannot be a practical preclinical tool to go to FDA or any regulatory agency. Hey, I have data I want to show you, because the model is not best suited to answer them unless someone comes in and say, hey, I proved it, which is possible. It's just people need to kind of demonstrate data. So your question, I don't know how long it takes, but there's enough of interest from the field. We need more validation and more trust from everyone into these technologies.
Okay, cool. So you brought up a very good point. So you mentioned that one part of validating these organs and chip platforms is for many people to be able to repeat the same experiments, so what are the challenges right now that not many people can repeat it, or is it at all a challenge?
Yeah, that's a good point. So I think it comes from who's asking. So if someone from industry asking this, is it because the technology is so new, is out of norm of the standard of practice they do, so they don't want to adopt it immediately? Is it because they are more inclined to use services as opposed to that? Is someone from academia and someone who has never done this organ on chip before and has no engineering background and wants to do that? So I think it all comes from who's asking, who's the client who wants to do that. But part of inherent challenges, I think, with these technologies is that when you want to build a good model, which you want to validate it, it takes time. It requires patience. It requires a lot of training. Even for us, I mentioned in my kind of talk that generating some of those data, like the COPD plot that I was showing you, it's 1.5 years of work, just one plot. Very few people have the funds, time, and patience to just dedicate multiple personnel, multiple resources just for that. So it takes time. Some people don't want to spend that much time. Some people want something that is like, oh, as if it's an off the shelf, so I can pick it up and I can just put whatever stimulus I want put in there and I can quickly get the data, or I just get it and I put it as something very interesting and sexy. I can put it in a grant application or a paper, so it makes it exciting. So I think we need to get over some of the kind of naivety. We also need to be practical. At the same time, we need to see what people need to adapt it and are there resources for them. If a funding institution doesn't support high risk, high reward, if they don't support kind of lengthy kind of experiments, it's very challenging to justify that. So all those sort of things comes into the problem of adaptation, like how to adapt it. Now you want to go to a commercial source to purchase them. You might be able to buy the kind of the individual devices, but if the protocol you try and normally you pay a significant amount of money, you buy them and you try twice, 3 times, and it lost money and it lost resources, you're going to be so disappointed. You're going to lobby against it. So it's one of those things that requires good education, good support from funding agencies, and people knowing that, okay, I need to have engineering background. If not, I better partner up with someone who has engineering background, if they're pure biologists. Or if someone engineer wants to do that, but they don't want to do it in their house, they want to need to work with someone who's a biologist and has expertise to kind of guide them and go through the model development.
Okay, that's really cool. Very. Thank you very much. So I think we're running out of time. So and I know you have other commitments. So thank you very much again, Dr. Benam, for your presentation. So I'll let Human conclude the session.
Thank you so much again for your great presentation, Professor Benam, and also the audience that joined us today. And I encourage everybody to follow us for the upcoming Wednesday with Professor Mekouani from Western University. Thank you so much and have a great day. Bye-bye.
Thank you very much. Have a good day.